Hi everyone. Hope you're all keeping fine and safe in your places. Sure, all of us we are locked inside our house. Nowhere to move, no schools, no colleges during this lockdown period. Though we are locked, but no one can lock our brain. It's we who monitor ourselves. And it's good that we refresh our knowledge. And I have decided to conduct some classes during this period, and it will be continued according to the queries arised. Okay, in today's topic, we are going to see RT-PCR. RT-PCR. What is PCR? To understand better this RT-PCR, we should understand what is PCR. We have learned already and we have the previous knowledge. What is PCR? PCR, which means polymerase chain reaction. Polymerase chain reaction. What is the uses of this technique? It is a technique and it is used to amplify the multiple copies of DNA. So what is the uses of this PCR technique? To amplify the multiple copies of DNA. So the DNA, it is double strand DNA, and this DNA, it is denatured and made into single strand DNA. The process we call it is denaturation. Denaturation. It is done by applying the heat 90 degrees Celsius. And this double strand DNA, it is made now a single strand DNA, and it contains the two end, that is 5' end and 3' end. And now this single strand DNA, it is now can be amplified in this technique. So how it can be amplified? By the enzyme, we call it polymerase enzyme. So that is how the reaction works. Because the enzyme polymerase, it is directing the DNA to synthesize. The polymerase enzyme is synthesizing the DNA. How it is possible to synthesize DNA? With the help of the oligonucleotides. So what does it mean again, the oligonucleotide? It is a primer, short sequences. We call it primer. So this primer anneals with this DNA. So the DNA 5' end and 3' end. With that, the primer anneals to this single strand DNA, and what is directed? The enzyme polymerase. So what is the role of this primer? Maybe this DNA, which has got lengthy sequences, some 25 to 50, some lengthy sequences, and the primer has got short sequences, oligonucleotide sequences, that will be synthesizing or adding nucleotides one by one, like forming a chain. As we are adding the beads in the thread, it will form a long chain. So as it prolongs, it continued and formed a new strand which is complementary to this strand. So maybe we can draw it in a color that is formed new strand. So it is a primer we kept here, and the primer that is synthesized, which is complementary to this DNA, a new strand is formed, and this will be amplified in the next step. So it is the way now this single strand will form a double strand DNA, and then again it will be amplified, the same strand, it will be amplified into other two strands. So when this reaction is continued, there is exponential growth. So which means a chain reaction that shows magnanimous amplification, multiple copies of DNA is formed. That is the thing we can explain here. So this is the process we understand PCR. Now we come to understand what is RT-PCR. RT, which means reverse transcription. Reverse transcription. For better understanding, still we can recall the central dogma of molecular biology. What it says, the central dogma of molecular biology, it is that DNA converts into RNA, RNA converts into protein. So, if the DNA is making its own copy, same copy, we call it a replication. If the DNA is making RNA, we call it is transcription. If the RNA is converting to protein, we call it is translation. Now, this is the normal process occurs in the eukaryotics. In the eukaryotic cell. Eukaryotics, which means both plants and animals. So these plants and animals, they have true nucleus and it has got this double strand DNA in each cells of the organisms. But in some other organisms like bacteria and virus, they have either single strand DNA or single strand RNA. Only few viruses, they have this double strand RNA. But most of the viruses, they have this single strand RNA or single strand DNA. Okay, for example, now all over the world is threatened due to the pandemic disease coronavirus. Now this coronavirus too has inside, just we think that is a virus and it has got only the RNA strand, that is single strand, it is single strand. Now, to detect the presence of this genetic material, that is RNA present in the human body, we need to run this technique. Which technique? PCR technique. But we understand these are PCR technique, we need to have single strand DNA. But in the virus, what it has? It has got RNA strand. But this RNA strand cannot be amplified in the technique. So, to change into a DNA, to make the RNA into DNA, we need a reverse transcription. So, that is the thing. Now, it does. The RNA that is inside these viruses, it undergoes for the reverse transcription, RT, which means from this RNA, it goes back to this DNA. Changing the RNA strand is converting into DNA, which means reverse transcription. And how it is done? It is done or it is catalyzed by the enzyme called reverse transcriptase. Reverse transcriptase. This is the enzyme catalyzing the reaction. So, for example, now we understand the process. the RNA that is removed or taken from the virus has got single strand, also has got 2 end, 3' end and 5' end. Now this single strand RNA, what it does, it will convert into DNA. How it can convert? By using the primer. By using the primer. And the enzyme, what enzyme? The enzyme reverse transcriptase. Reverse transcriptase is the primer, which is a short oligonucleotide, and the enzyme that is directing to synthesize a new strand with the help of this primer. So the primer anneals to this RNA, because now this RNA has got the sequences. Like for example, now you should understand what is sequences. Sequences, which means either DNA or either RNA or DNA, both have got sequences. In RNA, it has got the sequences, 4 sequences, that is A, U, G, C. And DNA, it has got the sequences A, T, G, C. In DNA, it is T. This T is replaced by the U. In RNA, there is no thymine, only uracil. So these are the sequences that is normally present in the DNA and RNA. But now, the sequences that is present in the RNA, coronavirus, already it is known, found out by China. They have given the sequences, what type of sequences are presented in the coronavirus. So, according to the sequences that is present in the virus, now many companies and different ways, they are providing the kit, that is this RT-PCR kit. Kit that contains the sequences. So, now the primers are designed according to the sequences that can match to the sequences. Now, this primer also have the sequences. This will detect that particular sequences where they are present. So, if these primers they are matching to these sequences, for example, sequences like A, U, G, T, sorry, here it is again A. So, those sequences it will be matching to the primer. So, it will form a complementary strand, that is here primer, then the new strand, it is extended in this way. Okay, so this is the newly synthesized DNA, which is complementary to this RNA strand. So now this is the DNA, we call it cDNA. Why? Because the sequences which is complementary to the sequences. Maybe the sequences that is synthesized is matching to the sequences that is present in the mRNA. So that's why we call it, it is cDNA. Now already the new strand is synthesized. The RNA, it is converted or made into new DNA. Now this DNA, it is going to act as a template, which means the strand that we are going to use to multiply or to make the multiple copies of DNA is going to act that DNA. So which means this cDNA can be used in the PCR technique. So which means now we direct PCR technique. So which means this is the strand that is synthesized. We are taking to PCR technique cDNA. And then from here 3' end, here 5' end, here 5' end, 3' end. So now these sequences again we take it to the PCR and the same process will continue. Again what process will continue? Denaturation will not occur, but now already the single strand is formed and then annealing will take place. Annealing means attaching this primer and making the synthesis of new strand, and then double strand DNA will be formed. So once in one step suppose one single strand, if it is making 2 copies, and then the second step or the second cycle, those two DNAs will form 4 copies, and then 4 copies will form 8, and then 16, 52. It will just go on and multiply number of copies. So just to say in simple way to understand, having like 1 copy and making Xerox. Having 1 copy, we can make multiple copies of Xerox, as many as number we want, is like that. Having the small amount of that DNA, we can make number of copies. Why? Now all over the world, especially in whole of India, in all the states, they are using this technique to detect the coronavirus in the patients. The technique they are using RT-PCR. There is another technique also. Another technique for that is used. One is RT-PCR. Another one is rapid test. These are the 2 tests we are using to detect this coronavirus presence in the patient's blood. Rapid test, we use the sample blood. We use the sample blood, but for RT-PCR, we take the sample from different solutions or the mucous substances, like from the nose or from the trachea or from the throat. We can take the sample that contains in those areas, because these viruses, they are able to survive those respiratory tract. So those mucous substances or sputum or any solution, thick solution that is present in the nasal pharynges, that can be taken using swab, like a cotton, how we can call it, the swab, buds, cotton buds, that we can apply and take those substances, and then we add different type of chemicals to separate this particular RNA strand from those samples. So again, now this simple strand, that RNA, they will take it to reverse transcription process and convert into a DNA, that we call it cDNA. And then the cDNA, when it is formed, then directly the PCR technique will be continued. PCR technique will be continued. So this is how they make the multiple copies. Maybe the sample that we are taking from the patients contain very small amount, but with that small amount, we can make number of copies, and to identify whether those number of copies have formed, we also identify this PCR technique. So at the end of the PCR technique, in 3 cycles, even we can make number of copies, and then we allow it to run 35 cycles. 35 cycles, we can make 35 millions of DNA. If the virus have affected the patient's blood, and those viruses transcribes, reverse transcription process, if it is transcribing the DNA, sure, those DNAs can be amplified through this technique. So the number of DNAs. If the number of DNAs have been amplified, it can be detected also, and it can be measured quantitatively. How it is possible? By applying a dye, a fluorescence dye called ethidium bromide. Ethidium bromide. Then another dye, we call it P32. So these 2 dye can be used. These are the fluorescents. Now suppose if they have formed 2 strands, these 2 strands, what forms? The sequences in the DNA, and then the newly formed DNA here, if they are complementary or matching, the dye will bind. The dye will bind here. The dye will bind when we apply. The more color is formed, the more copies have formed. That's the thing we can understand. Why? Because the color, the dye, the fluorescence, when we apply, it binds in those complementary sequences between those two strands, and they emit that color. They emit the color. Each fluorescence binding places, there will be color produced. Those fluorescence color produced. So with that, we are able to quantify how many numbers of viruses have been presented, or the DNA genetic material of the viruses have been produced, that we can be amplified. Suppose if only less number of copies have been amplified, and we find only the color formation only in few places or in few strands only. So that is the end of the identification. So this is how the RT-PCR technique is used in laboratory purpose. Actually, this RT-PCR is not a diagnostic test, but it is a research test. But now at present there is no other go, so we are using this technique, and all over the world they are using this technique, especially in India they are using the technique to find the coronavirus presence in the patients. Why we also focus to study? We as the students of microbiology and chemistry students and biochemistry students, we know the technique, but to understand more deeply together with these related diseases, how the term is going or the technique is conducted, we understand better. So here I stop. If there is any queries, you raise your queries and send to me my mail ID, and the assignment is just to raise your doubts. At least each student raise 2 queries or 3 or maximum 5. How many you can make it? At least make 2 queries. That will be greater exercise that you are doing in the house. Listen and just raise your questions, and if there is possibilities in next classes, sure we will see. Thank you.